Good afternoon. My name is Austin Campbell, and the topic I've chosen to cover for this assignment is the anatomy and enzymology of fat absorption. Before we get started, here is my napkin with my name and date on it for the assignment verification. So let's get started. First, I would like to start by briefly discussing the GI tract and the route of travel that foods and fats take throughout the digestive system. Things start out when food enters the mouth and mechanical digestion or chewing begins. Breaking up our food with our teeth may seem mundane, but is a very important first step in the digestion of food. Additionally, in the mouth, lingual lipase is introduced to begin the chemical digestion of fats. Simultaneously with the mechanical digestion. And lipases are enzymes that break down lipids into smaller lipids that are more manageable, and like free fatty acids. After chewing, food is swallowed, where it travels down the esophagus into the stomach. And then here in the stomach, some gastric lipase is introduced, but doesn't do too much in the big picture compared to other lipases. In the stomach, the food now becomes chyme, and continues on into the small intestine. And the small intestine, which is broken down into three sections, is where most of the action occurs regarding lipids and fats. The three sections of the small intestine are the duodenum, the jejunum, and the ileum. And in the duodenum, pancreatic lipase is introduced, and this is the main enzyme that converts triglycerides and phospholipids into monoglycerides and fatty acids. The fats are moved throughout the small intestine and digested and absorbed throughout it until what is left of the food reaches the large intestine. We won't discuss a whole lot of that because, as I mentioned before, the majority of the digestion and absorption has already occurred by the time the food reaches the large intestine, in terms of fats at least. Here are some visual aids to represent what I've mentioned in the last slide. As you can see, it displays the GI tract on the left, and more specifically on the right, we have the small intestine and its three components, the duodenum, jejunum, and ileum. Pretty basic stuff. And that brings us to our next slide, which dives a little bit deeper into the anatomy of the small intestine and what makes it so great at digesting fats and lipids. So food is moved throughout the small intestine by the movement created by smooth muscle contractions, which is done in a controlled manner to maximize absorption and so as to not overload the intestines. For example, the pyloric sphincter only allows a little bit of chyme through at a time into the small intestines so that the load can be managed. The small intestine plays a role in secretion, digestion, and absorption. And in order to be effective at each of those three processes, it has an increased surface area compared to the rest of the digestive tract. And this kind of occurs, this increased surface area is located specifically on the mucosal layer facing the lumen of the small intestine. The mucosal layer is covered with these finger-like projections and concavities called villi and crypts, respectively. And furthermore, the villi are lined with enterocytes on the brush border that have even smaller projections on the enterocytes called microvilli. And this picture kind of displays the villi and the crypts that increase the surface area, as well as the brush border that's created by the microvilli on the enterocytes lining the small intestine. Now we're going to talk about the digestion of fats and lipids. As I'm sure you know by now, at this point of the course, the lipids are not water soluble, and so they must be broken down into a number of steps and ways in order to be absorbed by the body. I mentioned before that some lipase activity occurs prior to the small intestine, such as lingual and gastric lipase, but for the most part, lipids are still in large clusters called droplets when they enter the duodenum, and they have to be broken down even further. So to combat these large droplets, the liver secretes bile salts that emulsify the lipid droplets and essentially get them into a solution and break them down into smaller and more soluble droplets that have a higher surface area comparatively total, and then these can be effectively reached by the enzymes that are present. Those enzymes are mostly lipase and phospholipase, which can now enzymatically break down the lipids, but they're aided by colipase, which is an enzyme that kind of breaks or displaces the bile salts that create this bile salt complex with lipids, and it displaces them so that the other enzymes can get to the core of the bile salt and lipid complex that's created and break those down. Once these lipids are broken down, they're then packaged into small structures called micelles that are able to enter the brush border and cross the brush border membrane for absorption. And that is kind of displayed in this picture. The slide shows the structures created by the bile salts when they emulsify the lipids, and also the micelles that are created to aid in crossing the brush border. And like I said, the colipase moves some of the bile salts out of the way in that top complex, so the lipase and phospholipase can reach the core of the bile salt lipid complex, and then the bottom free fatty acids and cholesterol are packaged inside of micelles to help them cross the brush border. Brings us to slide 7, where finally we will now talk about the absorption of fats. Some fats, such as like fatty acids and monoglycerides, are able to simply diffuse across the brush border because they're small enough. However, larger molecules like cholesterol definitely require transport proteins. And once inside the enterocyte, the fatty acids and monoglycerides reconfigure back into triglycerides, which seems counterintuitive because we broke them apart and then we put them back together. And then these triglycerides further combine with cholesterol and proteins into complexes called chylomicrons. The chylomicrons are packaged up by the Golgi, and they leave the cell via exocytosis from vesicles in the enterocytes. And once they exit the cell, they need to enter the lymphatic system. In order to do so, the chylomicrons use lacteals, which are special transport vesicles, into the lymphatic system. And once they're in there, they're able to get into the blood. Some fatty acids, if sufficiently short enough, they can simply diffuse through the membranes into the blood, and they circulate as free fatty acids. And this slide simply shows the structure of a chylomicron, which again is a packaged complex of proteins and lipids that travels throughout the body so that cells can take and use its components. That concludes the presentation on my anatomy and enzymology of fat absorption. Thank you for your time.